Today we're going to do a quick review of cell communication, cell cycle, and cancer. Cell-to-cell communication allows the trillions of cells in a multicellular organism to communicate and to coordinate their activities. Now, this isn't just important to multicellular organisms, it's also important to unicellular organisms. Biologists have discovered universal mechanisms of cellular regulation involving the same small set of cell signaling mechanisms. The ubiquity of these mechanisms provides additional evidence for the evolutionary relatedness of all life. Now, when you're looking at the basic forms of communication, you have local regulation and local signaling, and you have long-distance signaling. So autocrine signaling is when a cell signals itself to respond to something. Juxtacrine is when you have direct contact between cells, maybe through some sort of cell junction, gap junction, plasmodesmata, membrane-bound surface receptors. Paracrine signaling is when animals or whatever cells are releasing signaling molecules into the extracellular fluid around the cells, affecting nearby cells, or the local regulator function. And then you have a very particular type of that in neurons between the synapse or across the synaptic cleft. So that would still be a chemical communication over a local area, but because it's a specialized cell, it has its own name. In long-distance regulation, you have to travel through some sort of material, most often through the bloodstream, and those hormones are released by endocrine or exocrine cells to affect other parts of the body. Endocrine is going within the fluids, whereas exocrine is going outside of the body or into a body cavity. Well, what happens when a cell encounters one of these secreted signals, whether they're local or long distance? Well, the first thing is the signal must be recognized and bound to a receptor molecule. The signal, the ligand, is received by the receptor or the protein molecule that is picking up that signal. That signal is then passed through the cell into the rest of the body, or into the rest of the cell, I should say, through the process of transduction. That means there's going to be relay molecules, be they proteins like kinase or secondary messengers like CAMP, that will pass the signal from the membrane to whatever response there is. The response will typically be either the regulation of something that is already existing, like an mRNA or a protein, or the response will be the increase or inhibition of transcription as a transcription factor, causing the creation of mRNA or the inhibition of that creation, leading to long-term change. When you actually look at reception, we find that there are many different types of receptors. The one that is different from the rest is the so-called intracellular or nuclear receptors. These are ones that have ligands that are nonpolar, so they don't have membrane-bound receptors, and they don't have transduction. The ligand binds to the receptor protein and directly causes change. So that's only going to happen if you have something like a lipid ligand or maybe a gas. The other ones are different based upon the process of how they complete, but they're all membrane-bound, so they're polar. So ligand-gated ion channels or voltage-gated ion channels, you're going to have either a ligand, a molecule, or electricity that causes the binding, that opens an ion channel and allows the flow of things like calcium, sodium, potassium, what have you. Those are extremely common in things like the nervous system, looking at the sodium channels and the potassium channels that allow for action transport. G-protein coupled receptors are probably one of the larger families of receptors. It's called G-protein because it uses GTP. The molecules will trigger a cascade, be it phosphorylation cascade or secondary messengers to create cellular action. Your muscles, for example, use a lot of these. Kinase-linked receptors, these are ones that are formed as a two-part structure, and when the ligand binds, the kinase-linked receptors come together to form a dimer that is capable of autophosphorylating. It's able to transfer its own ATP and phosphorylate multiple pathways at once. So when you look at those diseases that have lots of effects, say like diabetes and the insulin response, you're likely looking at a kinase-linked receptor. Once the signal has been received, however it has been received, transduction is typically a multi-step pathway that greatly amplifies the signal. If some molecules in the pathway transmit the signal to multiple molecules at the next step, we'll see that broader amplification and multiplicity of a signal. Now, typically, you're either going to activate some sort of phosphorylation cascade, where you're going to have kinases, which are any enzymes capable of transferring phosphate groups, phosphorylating other molecules, and that will continue so long as the signal is being sent. That has to be able to be stopped, and that's where those PP molecules in, or those protein phosphatases, those will remove the ATP energy, the phosphate group, and inactivate the kinase to stop the response. Other times you're going to have secondary messengers, which are just non-proteinous signaling molecules like CAMP or calcium or DAG or IP3. All of these then can work with kinases to send a signal along to initiate a cellular response. The response is either going to be the regulation of an existing molecule like a protein or an mRNA, or it will work as a transcription factor and control the inhibition or production of mRNA to provoke a long-term response. But ultimately, that's really the only real option here. You're looking at proteins or mRNA or some other type of nucleic acid regulation leading to change. Now, when you look at these responses, we find that they're tied into two main types of response. You have positive feedback loops and negative feedback loops. In a positive feedback loops, it's enhancing, so more gives you more or less gives you less. Those are relatively uncommon in the body, but they do exist. Negative feedback loops are much more common as they are homeostatic balancers. They're maintaining an equilibrium. More gives you less, less gives you more. So an animal is a regulator for a particular variable if it uses an internal control. So it could be something like heat. The so-called warm-blooded or endothermic organisms are able to maintain a balanced body temperature through thermoregulation. And that is going to be an essential component of feedback control and homeostasis in many animals and organisms. The endocrine and the nervous system are the two systems that communicate the most. Obviously, both of these are capable of long-distance signaling. The endocrine does it obviously through endocrine signaling through the bloodstream, where the nervous uses electrical signals to pass signals throughout the body, and then chemical signals to pass signals across a synapse. But these two systems work together to control and to interact. So these hormones and these neurotransmitters may reach all throughout the body, but only specific target cells have the receptors that enable a response. Feedback is just extremely common within these systems. You could have negative feedback loops, like with pH. So as the pH drops in the duodenum will cause the release of secretin. That will then cause bicarbonate to be released, which will cause the pH to go back up. So that would be a negative feedback loop. Oxytocin is one that does positive, so as the baby suckles, oxytocin is released, causing muscles in the mammary glands to release milk. The more milk that comes out, the more the baby suckles again and again and again until the baby stops suckling. The same type of reaction can be seen with oxytocin and contractions in birth. So just because a hormone has one job does not mean that's the only job that it has. Glucose homeostasis is one of the most common examples of a hormone pathway, and here we can see that these pathways are typically antagonistic. You have two hormones, insulin and glucagon, that work together to balance out the blood glucose. So insulin causes blood glucose to fall, glucagon causes blood glucose to rise, to be released into the bloodstream, and these work together. So someone who's type 1 diabetic doesn't produce insulin, or doesn't produce enough insulin. Or somebody who's type 2 diabetic has been overexposed to sugars for so long that the body no longer responds to insulin. So this is just one of those most common examples of a metabolic balance and those simple hormone pathways that are part of the homeostatic control system. Now, we do need to realize that these hormones are being released throughout the bloodstream. They're going all over the body. So whether the response occurs in the nucleus or the cytoplasm, it's fine-tuned at multiple points. There's numerous steps for control. So epinephrine here, for example, is going to 3 different types of cells, and they either have different types of receptors or they have different types of intracellular proteins, and those mean that there's going to be a different response. So when you look at these systems that travel throughout the body, we need to realize that these systems are targeted and controlled. Homeostasis is maintained throughout. Now, sensing and communication is not just a multicellular thing. Bacteria use it in a wide variety of ways. One of the more common examples is quorum sensing. This is a bacterial gene expression system that is correlated to population density. So basically, transmission is dependent on autoinducers. They signal, there's autocrine signaling to release production and accumulation of molecules. Those molecules are released by the bacteria and they accumulate, and that accumulation is directly proportional to cell density. So as the organisms recognize more of self around or more of the same species, they will alter their responses. And we can see that example here with luciferase. So quorum sensing, the bacteria produces its own identifiable molecules that are species specific. when the recognition is activated enough, it can cause changes in virulence and other behaviors like biofilm production or bioluminescence. This is also, like I said, with virulence, why certain diseases, if you get few enough of these bacteria or whatever that may cause disease, something like cholera, you may have no effect. But when the population is high enough, they become virulent and it becomes damaging. Plants also use communication in a wide variety of ways to work within the multicellular organism. One example of this is plant tropisms, where they bend towards or away from a stimuli. Most common example of this, obviously, is phototropism. So any growth response that results in the curvature of the whole plant organ towards or away from a stimuli is a tropism. This is as close as plants get to taxis and kinesis of animals and those orientation behaviors. And this is very different from things like morphogenesis, which we'll talk about in the behavior section. When you look at animal cells, you first need to realize that animals are made of many different types of cells. In mammals, the embryonic cells are totipotent through the 8-cell stage, which is much longer than in many other animals. The totipotence of the very early cells is not fully equivalent in a normal embryo. Their totipotency, when isolated, likely means that the cells can regulate their response or at least regulate their fate in response to the embryonic environment. Once the 16-cell stage is reached, mammal cells are determined to form the tropoblast, or the inner cell mass, so they lose some of that potential and become pluripotent. So embryonic stem cells can become any type of cell within the body. So identical twins can develop when embryonic cells become separated, for example. But those cells differentiate and they are changed over time. A good example of this is obviously stem cells can become any part of your body. But once you're an adult, you don't have those pluripotent stem cells anymore. At best, you have multipotent ones like the hematopoietic stem cells in your bone marrow, which can become any of the many types of blood cell, be they platelets or the multiple varieties of white blood cells or red blood cells. So those cells, those pluripotent and totipotent cells, they must develop over time into the variety of tissues of the body. The first major determiner of this is cytoplasmic determinants. The molecules within the fertilized egg are not uniformly distributed. It's not homogeneous. They are different from area to area. And when the cell goes through mitotic cell division, different types of chemicals, different amounts of molecules and products will be available. And that will be the earliest step of determining the eventual fate of the cell. Once the cells have diversified enough that they have one type of control molecule, they begin to induce nearby cells to follow their determination path. So maybe you have a precursor cell that is being cytoplasmically determined to become a muscle cell, and it's going to, say, be a cardiac muscle cell. Then it's going to try to cause the other cells around it to also become cardiac muscle cells and to form the heart. So you have cytoplasmic determination leading to induction by nearby cells. This is a combination of autocrine and juxtacrine and working together to create these changes. The next step is going to be that precursor cell is going to have the alteration of their genes. So differential gene expression results from differential gene regulation in different cell types. So during embryonic development, cells become visibly different in structure and function. They do this through regulatory genes. So our cardiac muscle cell, for example, is going to have to turn on the MyoD regulatory gene, as that is the one that controls all of the other transcription-producing genes of muscle-specific products. So the cytoplasmic determinants will induce the activation of master regulatory genes. Those will then determine the cell to be a myoblast, or a precursor to a muscle, a blast. So you're looking at the broader structure that is going to be capable of forming muscle tissue. The master regulatory gene will act as a transcription factor, activating the other genes needed to produce a muscle, and eventually creating a fully differentiated cell. So the cell becomes determined, which is the chemical steps that determines its identity and determines its cell fate. But then when it actually produces the products that are different, that is the differentiation. So cells can be differentiated into all sorts of things, but how do you look at responses that are outside of that? How do you look at the nervous system, for example, and look at how responses are received as a broader multicellular organism? Well, the nervous system is a great example of that. We're going to look at that over the next couple of slides. So first off, you have to receive a signal. You have to receive communication or some sort of stimuli. That is then going to be picked up by receptors. That could be directly a neuron or could be some other form of receptor, like the receptor shown here inside your ear that allows you to hear. That is going to create an electrical signal that is passed through the neuron. And you can see that in the chart on the right. The action potential of that electrical signal that is generated, the more often the signal is received, the stronger the stimuli. The action potentials are always the same. That signal is going to be either inhibitory or excitatory. The inhibitors are going to cause hyperpolarization. They're going to cause stimuli that's going to lower the membrane potential. It's going to go more negative. That will not provoke a response. Signals that excite are going to cause depolarization. But if the depolarization does not reach threshold, then no action potential will be generated. If threshold is reached, the sodium channels will open, creating that first component of the action potential. And then once it passes 0, the potassium gates will open, which will begin to turn that process around. And then when it drops below resting potential, that hyperpolarization must be reset using a sodium-potassium pump. When action potentials along sensory neurons reach the brain, circuits of neurons process this input to generate the perception of stimuli. Perceptions, be it color, smell, sound, taste, these are constructions formed in the brain that do not exist outside it. But that electrical signal is passed through the neuron and causes action potential and change. When that electrical signal reaches the synapse, it's going to open a voltage-gated ion channel of calcium. That calcium will fuse or move into the neuron, and that will then cause the synaptic vesicles to fuse with the membrane to release the neurotransmitters. The neurotransmitters will diffuse across the synaptic cleft and then act as a ligand for a ligand-gated ion channel to generate a new action potential in the. When this cell starts off the cell cycle, it has one copy of its genome. It has one chromosome of each type. During the S phase of the cell cycle, during interphase, it's going to duplicate. So the DNA is going to double. But because it is a sister chromatid, it is a copy of the chromosome, there is still only one chromosome. So to differentiate this, we say that there is one chromosome that is single-stranded, and now there is one chromosome that is double-stranded. At the end of mitosis, those sister chromatids will be separated, and you will have one chromosome that is single-stranded in each cell. So mitosis is non-reductive because the number of chromosomes never changes. Now the actual chromosome structure that we see here only exists when the chromosomes are being moved around. It's a highly condensed form of chromatin. So normally the DNA is diffuse into that broader euchromatin, heterochromatin mix. So the chromosome is only a temporary structure. Cells spend the majority of the cell cycle in interphase, and the purpose of interphase is for cell growth and preparation for mitosis and cytokinesis. By the end of interphase, the cell has 2 full sets of DNA and is large enough to begin the division process. Interphase is divided into 3 phases. You have G1, gap 1, where the cell grows and synthesizes proteins. During the S phase, chromosomes replicate and divide to form identical sister chromatids. And during the G2 phase, the cell is going to continue to grow and produce the proteins necessary for cell division. Now, there are times when the cell needs to leave the cell cycle and quit dividing. That is going to be the G0 phase. It's a non-dividing differentiated set. This may be a temporary resting period, like in liver cells, where the G0, they exist in G0 but can be called back by external cues, or something more like the nerve and muscle cells, which are so highly specialized that they're not capable of dividing anymore and are arrested in G0, can never divide again. In late interphase, the chromosomes have been duplicated but are not condensed. A nuclear membrane bounds a nucleus which contains 1 or more nucleoli. The centrosome is replicated to form 2 centrosomes, and in animal cells this means 2 centrioles. So what really happens in mitosis? Well, mitosis is the division of the nucleus, so we're going to ignore all the rest of the organelles right now. In prophase, we see that those centrosomes are beginning to release spindle fibers, the nuclear membrane is beginning to break down, the chromosomes are beginning to condense. It's really prepping the rest of the process. And in pro-metaphase, the spindle fibers have attached to the chromosomes, and you now have created the kinetochore, those movement centers. That begins to move the chromosomes around, and when they get to the middle of the cell, that is the metaphase point. At metaphase, the proteins that hold the chromosomes together at the centromere are broken, and you move into anaphase, and the sister chromatids separate and move to either end of the cell. Telophase is merely the opposite of prophase, so you have the rebuilding of the nuclear membrane, the decondension of the chromosomes, the nucleoli reforms, things like that. And then after telekinesis, you have cytokinesis. Cytokinesis is separate from mitosis, but is shown here. Cytokinesis is the division of all things except the nucleus. So all of the organelles and everything are divided in the process of cytokinesis. The mitotic spindle distributes chromosomes to daughter cells. It's fibers composed of microtubules and associated proteins, and it's the major driving force of mitosis. As the spindle assembles during prophase, the elements come from the partial disassembly by other microtubules of the cytoskeleton, and those spindle fibers polymerize by incorporating these tubulins and begin to move the chromosomes around. When you actually look at the process, we see that the motor proteins inside the kinetochore are moving the chromosomes along this microtubule and breaking it down as it passes. So this movement is essential for the rest of this process, and if it fails, you have non-disjunction and polyploidy mutations. In animal cells, cytokinesis occurs by a process called cleavage. The first sign of cleavage is the appearance of a cleavage furrow in the cell surface near the old metaphase plate. On the cytoplasmic side of the cleavage furrow is a contractile ring of actin and motor proteins like myosin. The contraction of this ring pinches the cell in two. Cytokinesis in plants is completely different. During telophase, vesicles from the Golgi apparatus move along microtubules to the middle of the cell where they coalesce to form a cell plate. The cell wall materials are carried by those vesicles and collect as it grows. The plate enlarges until it fuses with the plasma membrane, and the contents form a new cell wall between the daughter cells. The timing and rate of cell division in different parts of animal or plant cells are crucial for normal growth, development, and maintenance. The frequency of this division varies with cell type. Human cells, like skin cells, may divide frequently, but liver cells may only do it every now and then, and nerve and muscle cells never do it once they are mature. So investigating these processes have made us realize that cytoplasmic signals drive the cell's cycle, and we find that there are three key checkpoints that control this process. One is at G1, one is at G2, and one is at M. This cyclical operation triggers and coordinates key events in the cell cycle, and so it's basically a built-in clock, but this clock is also regulated by external adjustments and internal controls. The internal controls, aka promoting factors, are going to be signals that are released from within the cell to cause change in the cell cycle. One of the most common examples of this is the combination between CDK and cyclin. Cyclin is a control molecule that is broken down as needed, and we find that there are many different types of cyclin that control what phase of the cell is in. But the cyclin-dependent kinase, which is kinase and it's anything capable of phosphorylation, is going to be maintaining a constant level. But it only phosphorylates when the cyclin is attached because it is cyclin-dependent. One of the more common examples of this is MPF, which we'll look at in just a second. MPF, or the mitosis promoting factor or maturation promoting factor, there's a variety of names, promotes mitosis by phosphorylating a variety of proteins. It acts both directly as a kinase and indirectly by activating other kinases. It stimulates the fragmentation of the nuclear envelope. It also contributes to chromosomal condensation and spindle formation. The non-cyclin part of MPF, the CDK, persists in an inactive form until it associates with the cyclin that is synthesized during a variety of phases to do the actual work of the cell. The M phase checkpoint ensures that the kinetochores of all the chromosomes are properly attached to the spindle at the metaphase plate before anaphase. In this case, the regulatory molecule is not a cyclin CDK, but a complex made up of several proteins. Once activated, this sets off a chain of molecular events that activates the separase enzyme. The separase enzyme cleaves the cohesins and allows the sister chromatids to separate. How does the APCC do this? Well, it looks like it has a different function than CDK. It uses ubiquitin to destroy securin, the actual molecule that prevents the activation of separase. And so not all molecules are going to be regulated the same way. DNA damage can and will happen in many cells of the body during a person's lifetime, for example, UV rays from the sun. So cells must be able to deal with this damage, fixing it if possible, preventing the cell division if not. The key to the damage is going to be these tumor suppressor genes like p53, which are described as the guardian of the genome. p53 works on multiple levels to ensure that cells don't pass on damaged DNA. First, it stops the cell cycle at the G1 checkpoint by producing CDK inhibitors. That's going to block the activity, giving time for DNA repair, which p53's second job is to activate DNA repair enzymes. If the DNA damage is not fixable, p53 will play its third and final role, triggering programmed cell death so damaged DNA is not passed on. By ensuring that cells don't divide when their DNA is damaged, p53 prevents mutations from being passed on. When p53 is defective or missing, that can then lead to an accumulation of mutations, possibly leading to cancer. Particularly important for mammalian cells are growth factors. These are going to be external controls released by certain cells that simulate other cells to divide. Researchers have discovered at least 50 different ones, and a good example of this is the platelet-derived growth factor. This is produced by blood platelets and is required for the division of fibroblasts. These connective tissues have receptors on their membranes and allow for the response to injury. We do find that there are two major control mechanisms for these growth factors. One is anchorage dependence. So to divide, the cells must be anchored to a substratum, typically the extracellular matrix of a tissue. The density-dependent inhibition is looking at the actual level of growth and the amount of organisms or the amount of cells within an area. Cultured cells normally divide until they form a single layer on the inner surface of the culture container, and if a gap is created, the cells will grow to fill that gap. But if you have something like cancer, tumors build because the density-dependent inhibition is not working, and they're able to spread through metastasis or metastasis because the anchorage dependence fails too. When you end up with uncontrolled cellular growth, that's how you have cancer. Now, more than one somatic mutation is generally needed to produce the changes characteristic of full-fledged cancer cells. If cancer results from an accumulation of mutations, and if mutations occur throughout life, then the longer we live, the more likely we are to develop cancer. The first sign of cancers is typically going to be some sort of polyp or small benign growth. That's, in this example, at least of colon cancer. The cells look normal but divide unusually frequently because of the loss of, in this case, tumor suppressor genes. Through gradual accumulation of mutations that activate oncogenes and knock out tumor suppressor genes, things like polyps can develop into a malignant tumor that can spread. This might be turning on RAS or turning off p53. About half a dozen DNA changes must occur for a cell to become fully cancerous. Because mutant tumor suppressor genes, for example, are usually recessive, mutations have to knock out both alleles. So it's very complicated when you actually try to look at how cancer develops. But it happens, and looking at those broader characteristics of cancer cells can teach us a lot about how we can try to treat it. At its most basic level, cancer results due to a failure of signaling regulation. This could be like in the example of RAS, where the RAS becomes deregulated and stays active even when the ligand is not present. Or it could be a defect in something like p53 on the right, and so the cycle inhibiting pathway is defective and you have no inhibition process. So it's not just one mutation, it's a series of mutations, and they're acting on multiple levels. So when you really think about it, we all have cancer in us as part of our cells because we all have genes, these proto-oncogenes, that regulate cell cycle. And when those cell cycle regulators are defunct or are overactive, that leads to difficulties. And cancer is one of those. There are many different types of cancer treatments, and the type you receive will depend upon the type of cancer you have. We can work to prevent cancer by early screening, like pap smears, or by getting vaccines for viruses that can cause cancer, like HPV and HBV. But hormone therapy, for example, might be more useful for something like breast tissue, where you have cancers that respond to estrogen. Surgery is going to be useful in a variety of situations, but may not always be effective. It depends upon the location of the tumor. Bone marrow transplantation is only going to be effective for blood-based cancers like leukemia. Chemotherapy, the problem, of course, is that you are adding toxins to your body, and those toxins can kill you. And they attack frequently dividing cells. So you're going to have issues with your intestines, you're going to have issue with your hair, but the goal is to kill the cancer before the treatment hurts the body too much. Radiation therapy is in the same situation as chemotherapy. It's all about how you target and how long, but radiation is inherently dangerous. Immunotherapy tries to use the immune system to fight cancer. Because think about it, your immune system is preventing cancer every day. If we can find ways to boost that, that might be a really good thing. Targeted therapy is really where we're going in the future, though, because targeted therapy looks at exactly the types of receptors, the types of transducers, the various pathways and genome of the cancer itself to create tailored treatments. And while that used to be incredibly hard to do, it's getting more and more affordable as we get through and we get further into analyzing the genome of all organisms. So cancer is preventable, at least certain types of cancer, and cancer treatment is getting better all the time. So it's not all doom and gloom. There is hope for the future.